Hi, and welcome to today's lecture, which is going to be all about Thomas Hunt Morgan. He was the fruit fly guy. You've kind of had a little bit of exposure to him already, but we're going to go way deeper into his discoveries and why he's so cool, and in my opinion, even cooler than Mendel. So, we've talked about this before, but Thomas Hunt Morgan is kind of famous for discovering sex-linked traits. He was the first to associate genes to a specific chromosome. So while Mendel was able to determine that, okay, these genes pass on, I don't know how, but I know that, there's dominant and recessive, and they don't seem to be tied together. They just independently assort all of this. Morgan was able to say, no, no, no. This gene is tied to this specific chromosome, and here's my proof. He used the fruit fly, Drosophila melanogaster, for his experiments, and so fruit flies to Morgan are as peas to Mendel. They were used for everything. You're going to get sick of fruit flies soon enough. So Morgan was really famous for this sex-linked traits, but it wasn't just that. He also was able to kind of bring to light a lot of new genetic information that has led to a number of other discoveries since then. So we've talked about genes found on chromosomes, we've talked about sex-linked and multiple alleles, but what we're going to get into is epistasis, linked genes, and gene mapping. Now, linked genes is the idea that genes get inherited together. So if you inherit, I don't know, let's say brown eyes, you would automatically inherit brown hair. That's not the case for humans, but for fruit flies, there are a number of linked genes. So say you inherit red eyes, you are guaranteed to inherit flat wings, something like that. Okay, so they get inherited together. That's the idea of linked genes. Gene mapping is the idea of being able to identify where on a chromosome a gene is located based on its location to a second gene. You're going to get a lot of practice with this, but just so that you kind of have an idea of what I'm talking about now. And then epistasis. Epistasis is the coolest, in my opinion, thing that you're going to learn about. So epistasis is this idea that you have multiple genes, but some genes can only be quote unquote turned on, seen if the first gene is there. This happens a lot in like fur color with animals. So let's say there's two genes, one for brown or black fur, and then the second one is for stripes. So you can only inherit stripes if you have a certain color in the first gene. We're going to get way more into this later, but epistasis is probably my favorite thing about Morgan's work. It's what I actually studied when I was in undergrad, so I like it. It's cool. So now let's get back to Morgan specifically. So the fruit fly, right? We've talked about why Mendel used peas. Now let's talk about why Morgan used fruit flies. Well, they're super common, they're really harmless, and you can get tons of offspring from one mating. The regeneration time is really short, so you don't have to wait forever for a pea plant to grow or for another organism to reproduce, right? You're talking like 2 weeks, you get hundreds of offspring, and there's only 4 chromosomes. 4 chromosomes is like nothing, okay? We have 46 chromosomes. Well, 46 pairs, I should say. 23 pairs, 46 chromosomes. But fruit flies only have 4 pairs, so there's only 8 total chromosomes. That's so simple. You can do so much work because there's only so many areas a gene can be found when there's only 4 pairs of chromosomes. Now, one thing about Morgan is that you're going to see that he uses a different type of annotation to talk about his genes and the alleles that he's looking at, whereas Mendel used capital letters and lowercase letters, Morgan didn't. And so first we need to talk about the 2 types that he talked about. Instead of dominant and recessive, Morgan called them wild type and mutant types. So a normal phenotype would be the wild type. It's it's what you would see in the wild, right? It's the most normal version. So red eyes, normal wings, like a yellow-brown body. That's the wild type, kind of like the first picture on the top left. That would be a wild type fruit fly. The mutant type would be white eyes, vestigial, dumpy, or no wings. Basically, any wing that isn't normal is a mutant, and then an ebony body. So the mutant type is anything that is not normal, right? Something that is a mutation. Major differentiations between male and female is male has sex combs, a rounded abdomen. You can see that at the bottom here, right? It's like rounded right here, whereas the female is a little bit more pointed. You have 5 segments. Females have 7. Females are typically larger, so when we look at fruit flies and do a fruit fly lab, you're going to be able to really identify these differences. And now, the notation that I was talking about. So when you write the alleles, when you're using Morgan's notation, you use a plus sign. I know, as if I had to give you one more thing, but Morgan was very specific in his notation, so we're going to also be specific in our notations. So you use a letter for the mutant phenotype. So, for example, if we're talking about wings, one of the most common mutant phenotypes of wings is vestigial. So if I were to notate vestigial wings, I would use VG. And you can see that at the bottom, right? Homozygous for vestigial wings. So the letters you use is for the mutant phenotype, so VG, vestigial. And then if you were the wild type, you would have a plus sign. So homozygous for the wild type, aka normal wings, would be VG plus VG plus. So this is notating that we're talking about wings, where the mutant type is vestigial, but in this case, because there's a plus sign, it's normal. It's the wild type. So homozygous for the wild type, for vestigial wings, which is kind of like backwards from what we were doing with Mendel, but that's okay. And then homozygous for vestigial, no plus sign, so we know it's vestigial, and then heterozygous. So again, this is just kind of another slide that says this. So normal alleles are usually the most common, designated by a plus symbol, and usually dominant. So the wild type would have a plus sign, whereas the mutant would be the letters. And if you were to notate wild type, you would use the mutant letters and then put a plus sign after it. So test crosses, we've talked about this before, but Morgan was crossing wild types with various mutant types to determine patterns of inheritance. And based on that, he was able to discover sex-linked traits. When he discovered sex-linked traits, he also was able to find linked genes, meaning that 2 genes were inherited together. And the way that he found that is because the results that he was getting just didn't match what was expected. So those Mendelian ratios that were working for everything else just weren't working here. And so Morgan was like, okay, something new is happening. Let's figure out what it is. And so this idea of linked genes. The genes carried on the same chromosome, and they're linked from parent to offspring. So they're inherited like single genes, right? They're inherited together. So in this case, if you're looking at the bottom picture here, A and B, oh, now I can't see it, but they're linked, so they would get inherited together, right? But A is up here and C is way over here, and they're not linked, so they would be independently inherited. Okay, so because A and B are so close, they would get inherited together because they're linked. But A and C are not, right? They're not on the same chromosome, so they're going to be inherited separately. Now, to add another layer, there's this idea of recombination. Recombination is the idea of typically linked genes that are inherited together get split and recombined so that they do not get inherited together. So this idea of recombination This is just the words that I was just saying, but now it's in front of you. So these are the alleles that are linked on the same chromosome, but they exchange genetic information in something called crossing over, and then they don't get inherited together anymore. They're called recombinants. And we're going to get into this more. We're going to practice with this, but this is just kind of your notes to show you where we're heading. Okay, so this is happening during meiosis, during prophase 1, and you've all learned about meiosis, but we're not going to go super deep into it. We're just going to talk about this idea of crossing over. And to give you a better picture of this, so this is this exchange of genetic information. And you can see this right here, right? You have the dark blue and the light blue. And then they kind of meet and they cross their genetic information. So if you had a linked gene that was on, this arm of the chromosome, when they swap, they're no longer being linked in their inheritance because they have a new allele there, right? It might be the same, but it might be different because you have different alleles, right? One from mom, one from dad. So crossing over is what leads to recombination. So this is just kind of like talking more about why we know that this happens. So this is all the idea that you are not getting the ratios that you are expecting to get from a dihybrid cross, right? If you had a homozygous strain for purple and for red flowers, and then you had the F1 generation, and you would expect to see all long pollen purple flowers, right? Because that's what would typically happen with the dominant trait. But then when you do the F2 generation by self-fertilization, you do not see the typical 9:3 to 3:1 ratio. Okay, and because we don't see that, we can assume that there's something funky going on, and it's probably due to linked genes. Okay, I have this kind of twice, I guess, but we'll go over it again. So just to recap, linked genes are genes that are close together on the same chromosome. When you have linked genes, the genes are inherited together. However, crossing over can happen, and that produces recombination, which breaks up those linked genes so they are not inherited together. And that's this new idea. And recombinants always look different than their parents, so you can easily tell what the recombinants are. A recombination frequency is the amount of recombination between the two genes. So you can figure out how frequent this recombination is going to happen based on the distance between the two genes. So if the genes are farther away, they're going to have a higher recombination frequency because it's going to happen more frequently, right? If they're farther away, it's easier to break off between those two genes so you get more recombinants. If the genes are closer together, then the recombination frequency is going to be much more limited because it's harder to break, have that crossing over happen in between those two genes that are really close together. And then you can obviously do math, right? Because everything we do kind of ties back to some form of math, right? And so this is calculating recombination frequency. So if you have recombinants, you add up the recombinants, divide by the total number of offspring. So recombinants and your normal, what you would expect to have if you had linked genes. So recombinants divided by the total number of offspring, multiply that by 100, and you get your recombination frequency. The higher that is, the further away the genes are. The lower it is, the closer together the genes are. And now, what you're going to be working on first is gene maps. So gene maps tell you where the gene is located on a chromosome. So this is ideal if you are trying to figure out how close together genes are, or if you want to figure out how easy it would be to have crossing over happen, anything like that. So there's two maps. There's the linkage maps, which is a chromosome map. It's very abstract. It's based on recombinant frequencies. and then physical maps where you're actually positioning genomic fragments. So you're literally putting fragments of the gene together to make this map. We're going to be focusing on linkage maps, this more abstract idea where you're just kind of determining where you think these genes would lie on a chromosome. We're not going to get into the really specific fragments. It's just a little bit too much for us. Okay, so talking about linkage maps. It's our first genetic map, and a new scientist, Alfred Sturtevant. He worked in Morgan's lab, and he observed that some pairs of genes don't segregate randomly, like what Mendel was saying. And so he actually proposed that genes were located on the same chromosome, and the variation in the strength of the linkage, right, determined how the genes were positioned on the chromosome. So he was kind of that guy that said, this is happening more frequently. I think the genes are further away. Or this is not happening very frequently at all, so the genes are probably close together. So you can thank this guy for what we're about to start studying. Now, I've been talking about these linkage maps and these chromosome maps, and I haven't really shown you what that means. And so this is what that really looks like. So you use your recombination frequencies to determine your relative location. So if you have a high recombination frequency, the genes are further away from each other. In this case, you could say gene A and gene B have a recombination frequency of 9.6, so they would be 9.6 map units away from each other. Okay, and so you can see that they're the furthest away. They have the highest rate of recombination. And then A and C have 8% recombination frequency, so they are 8 map units away from each other. Okay, so still kind of far, still relatively high. But then C and B only have a 2% recombination frequency, so it's unlikely that these guys go through recombination very frequently because the percentage is low and they're really close together on the chromosome. Okay, and then our last bit, well, in relation to this, is this Drosophila chromosome. So you can see on this chromosome, when we're looking at this chromosome, when we bring it out, okay, we have the Aristotelis, which is like their antennas almost, the wing gene, the body, the eye color, the wing length, and another bit of eye color, whether it's brown or purple would be your mutants here, okay? And you can see the distance, okay? So you have 13 map units, 48.5, 54.5, 67 map units, 104.5 map units. So you can tell that if you were looking at the difference between dumpy wings and brown eyes, it's likely that this would have some recombination happening because the distance is so far. But if you were looking at the black body versus purple eyes, that will likely get inherited together because it is so close together. It's unlikely that you would have crossing over happening there. Okay? So the further away, the easier it is to do recombination. So now, predicting outcomes of crosses with linked genes. Okay, so this is all about knowing the recombination frequency. Fruit. Okay, so if you're crossing this warty dull fruit with this smooth glossy fruit, you can see that this is a test cross because it's all homozygous recessive. And then you have your homozygous cucumber, I guess, and you have your gamete formations, okay? The only gamete formation you can have with this test cross cucumber is the homozygous recessive, but for your gamete formation, you can see that you have 2 non-recombinant gametes and then your recombinant gametes. And the way that you can tell that these are recombinants is by looking at this top part, the way that you display your genes. Again, this is a Morgan specific way to show your genes. you can tell that your non-recombinants are the ones that are on the same gene. So in this case, you have capital T and capital D on the top. That is a non-recombinant. Then on the bottom, you have lowercase t, lowercase d, non-recombinant. And you can see that right here, that these are non-recombinant gametes. And they make up the majority of your predicted frequency, right? But you also have your recombinants. And to figure out recombinants, you just swap the last two letters. So it would be capital T, lowercase d, which is not what you see here. And that is a recombinant. And then lowercase t, capital D, again, not what you see in the parent, it's the opposite. So that is a recombinant. And then based on this, you can tell that the recombinant frequency is 16% because 0.08 plus 0.08 is 0.16 times 100 is 16%. So the recombinant frequency of this is 16%. Pretty crazy. We're going to practice a lot, so don't worry. I know that this is kind of a lot of stuff happening right now, but we'll get there. Don't worry. In this same example, it is literally the same cucumbers, and it's again talking about the same stuff. So this is it's like taking these outcomes and just putting pictures with them. Okay, so you have your normal, what you would expect to see, and then you have your recombinants. Now, I was telling you earlier that the recombinants are always the ones that don't look like the parents. Well, in this case, that's exactly right, because your parent is warty and dull and smooth and glossy, right? And you see that on the previous slide, warty and dull, smooth and glossy. Your recombinants are the mashup of those, so warty and glossy and smooth and dull. So these are your recombinants, and in this case, we got 0.08% for your recombinants. If you add them all together, it's 16%. So it's all the same math, it's just that this is like a visual representation of what I was talking about. And then again, you are mapping with those recombinant frequencies. So this is this idea that you calculate the recombinant frequencies, and then you use that frequency to determine where those genes are. When you're making genetic maps, it's measured in map units. You may have heard me say map units previously in this lecture, and you're like, what is this crazy lady talking about? Well, map units, 1% recombinant frequency is one map unit. It's all 1 to 1 to 1, right? And centimorgans, because scientists love to name things after themselves. So recombinant frequency, if it's 1% recombinant frequency, it is one map unit and it is one centimorgan. It is all 1 to 1 to 1. It all equals the same thing. So no matter what language you're using, it all is the same exact number. It's just one more way to confuse you. And then a recombinant frequency, it can't exceed 50% because once you're at 50% or greater, you can't distinguish whether those genes are on the same or different chromosomes. So you kind of just, if your recombination frequency is over 50%, you have to say that you are unsure that the gene is linked or unlinked, because you can't prove one way or the other. And then with that, you can have double crossover, so you can underestimate the distance, which would make you say that it would be linked, but it's actually not, which is why we just don't mess with anything that's over 50%. This is again another image of this kind of mapping. So you have your genes. The further away you are, the more likely that recombination is going to happen. In this example, MU stands for map units, so you can see that A and B is 23 map units away, B and C is 34 map units away, and based on that information, you could figure out that A and C are 57 map units away just by adding up the distances between those two. So when you're doing this, you kind of have this idea that, again, if they're widely separated, they're going to often recombine. And so they will seem to assort independently. And so you would figure out if things are linked by testing the genes between whatever ones you are looking at. So in this example, genes A and C are so far apart that typically you have crossing over. So you can't determine if it's linked, but you would test their relation to B, which is in between the two. And because those are close enough to show linkage, then A and C must therefore also be linked because A and B are linked and B and C are linked. So even though crossing over happens super frequently, by doing other tests, you can determine if there is linkage there. Okay, and then finally, this is just another picture of a simplified map of the chromosome for Drosophila. We've seen this before, but just so that you have a better visual of it and you can see kind of the phenotypes of everything. We're going to get way more into this stuff, so it's good to have an idea of what's happening here.